it's really been breathtaking in recent years to see the rate of progress in China and the growth of science in China, both in terms of the scale of the science as well as the international impact. There's probably no country in the world that's strengthening its science as quickly as China is. And it's incredibly impressive to see. And it's incredibly impressive to see the amount of talent scientifically training in China. I only wish that I could have been there in person at the meeting, both because the hospitality is always amazing when I've been invited to meetings in China. It's incredibly interesting for me to be able to go. And plus, I've got a bunch of friends there at the meeting that I haven't seen in some years as a result of the pandemic. And so I'm looking forward to having dinner with all of you in person when we have an opportunity to see each other again. So let me just share my screen. Let's see. Okay, so assuming that you can see my screen now. So my laboratory studies the intrinsic mechanisms within stem cells that allows them to execute self-renewing divisions. Those are the divisions that allow stem cells to divide to make more stem cells. And that's how stem cells persist throughout life in your tissues. We've studied this particularly in the hematopoietic system as well in the nervous system. In addition to studying self-renewal mechanisms inside the stem cells, we also study the extrinsic mechanisms by which the niche or the microenvironment helps to maintain hematopoietic stem cells within the bone marrow. And then we study the ways in which all these mechanisms get hijacked by cancer cells to make tumors. So mammalian stem cell niches generally haven't, we talk a lot about niches or microenvironments for stem cells, but these microenvironments generally haven't been characterized at single cell resolution. And the hematopoietic system in many ways, as you know, has been a paradigm for understanding stem cell biology. And so my lab for the last 15 or 20 years, one of the things that we focused on is identifying this niche that maintains hematopoietic stem cells within the bone marrow. So the key question was which cells are producing the growth factors that helps to maintain the stem cells. And now that we've identified the niche, it's a cool time in the field because there's an opportunity to go beyond the growth factors and ask what else does a niche do? Okay. So the bone marrow is where most stem cells are located in adult humans. And most of the new blood cells that you form every day form in the bone marrow. So you have to make new blood cells every day to maintain your blood cell counts. But the bone marrow is not just a big bag of blood forming cells. There's also a complex network of blood vessels and stromal cells within the bone marrow. And when my lab started working in this area, very little had been done to characterize the stromal cells in the bone marrow. So blood flows into the bone marrow through these arterioles that are colored in green, and then it transitions through these transition zone vessels that have properties partly similar to arterioles and partly similar to the venous circulation. There's a specialized form of venous blood vessels in hematopoietic tissues, including the bone marrow, called sinusoids. So the blood flows into the bone marrow through these green arterioles, and then it transitions through the transition zone vessels into the sinusoids and then flows out of the bone marrow. And what I'm going to show you is that my lab discovered that the stem cells reside adjacent to these purple sinusoidal blood vessels, and that the key cells that provide the factors for hematopoietic stem cells also reside around these sinusoidal blood vessels. So our first clue of this was back in the early 2000s when Mark Keel and Omer Yilmaz did gene expression profiling on hematopoietic stem cells. Omer is now a professor at MIT. Mark Keel went on to start his own bioinformatics company called Genomenon. They were incredibly insightful in seeing a very complex pattern of gene expression in the hematopoietic stem cells, where they noticed that a single family of cell surface receptors called the SLAM family was differentially expressed among hematopoietic cells at different stages of the hierarchy. So there was one SLAM family member on in the stem cells. There was another one on in multipotent progenitors. There was a third one on in restricted progenitors, a fourth one on in differentiated cells. And they came to me one day and they said, we think that based on this pattern, we'll be able to improve the purification of hematopoietic stem cells just using these SLAM family markers. And I said, well, you know, these markers are never as good as they look by gene expression profiling. So I kind of discouraged them. I said, don't spend too much time on it. But they turned out to be exactly right. These markers dramatically improved and simplified the purification of hematopoietic stem cells. And to this day, these are the markers that people use to purify the stem cells. But the part that's relevant for this evening's talk is that, or I guess this morning's talk for you, is that this was the first time that it was possible to purify stem cells using a simple two-color combination of markers. And that made it possible to cut sections through hematopoietic tissues to see where the stem cells were located. And when we did that, we found that most of the hematopoietic stem cells were immediately adjacent to the sinusoidal blood vessels. So this white stain is panendothelial antigen. This is the lumen of the sinusoid where the blood flows. Giant yellow cell is a megakaryocyte. So when we saw that the stem cells were adjacent to the sinusoids, we proposed the existence of a perivascular niche. Now, at that point, there were very different ideas in the field about where the stem cells were and what the nature of the niche was. So this was an idea that it seemed like nobody thought could be right at the time. So as we continued to test this idea and the data kept coming back consistent with this model and inconsistent with the prevailing model at the time in the field. And Lei Ding came to my lab, also an outstanding scientist, now has his own laboratory at Columbia University where he's continuing to do great work on the niche. And he made a lot of mice that really changed this field. So he made reporter alleles where he knocked GFP or DS-RED into the loci that encode two known niche factors that were known to be important for stem cell maintenance. And the idea was there was really no good data on the expression patterns of these factors. So when we proposed that there was a perivascular niche, we were assuming that there would be perivascular cells that would make these factors that were required for stem cell maintenance. But nobody had any data on which cells were making those factors. So we thought with these reporter alleles, we could determine the expression pattern. And then he also made floxed alleles of these factors and then used Cre recombinase expressing alleles in all of the candidate niche cell populations that anybody had proposed. And he mated all of these Cre expressing alleles with each of these floxed niche alleles to conditionally delete each factor in every candidate niche cell population. And so this was an opportunity for us to both disprove our own model as well as testing all of the other models that were present in the field. So this is what Lei saw when he cut sections through the bone marrow of mice that were expressing GFP, green fluorescent protein, under the control of the stem cell factor locus. So the idea here was that the niche cells would glow green. And so this is a low magnification section through a femur, where this is bone in the bottom and bone on the top, and this is all bone marrow in the middle. So you can see that the green is in these little rings that are present throughout the bone marrow. In fact, these rings are the sinusoidal blood vessels. So we were seeing the stem cell factor exactly where we had seen the stem cells. And when we looked at higher magnification, we could see a very low level of stem cell factor in the endothelial cells that created the walls of these sinusoids. But the most of the stem cell factor was present in stromal cells that were around the outside of the blood vessels. Now, when we first saw this, we didn't know what these cells were because no one had ever characterized these cells. But Lei used the GFP as a marker to purify the cells. And by gene expression profiling, he discovered that he could distinguish these cells from other cells in the bone marrow based on their expression of full-length leptin receptor. And so Lei discovered a new stromal cell type within the bone marrow, these leptin receptor positive cells, that turn out to be the major source of factors for hematopoietic stem cell maintenance. And they make many of the growth factors that are critical within the bone marrow. So then Lei conditionally deleted both stem cell factor and CXCL12 in all of the candidate niche cell populations within the bone marrow that anyone had proposed were part of the niche. And for most of these cells, there was no effect on stem cells and no effect on hematopoiesis. And so that showed that these other cell types weren't the key source of niche factors. But when he deleted stem cell factor from either endothelial cells or from these leptin receptor positive cells, he got stem cell depletion. And going into this, everyone in the field thought that there would be a single niche cell type. But these data showed that there were two different redundant sources for niche factors in the bone marrow, endothelial cells and leptin receptor positive cells, suggesting that if we conditionally deleted stem cell factor in both cell types, that we should see a profound depletion of the stem cells. And so we did that experiment, and in fact, you do. All of the quiescent and all of the serially transplantable stem cells from the bone marrow disappear over time when we delete it in the endothelial cells and the leptin receptor positive cells. So that proved that the niche really was perivascular and that these cells were the critical source of growth factors. Even after that, there was still differing opinions in the literature about where the stem cells were located. And to be fair, in the 2005 paper, when we proposed the existence of a perivascular niche, Toshio Washita in my lab spent a month looking at thin sections through the bone marrow and counted what I think was a total of 36 hematopoietic stem cells. And he said that most of the stem cells were around the sinusoids, but then when other people did the experiment in other ways, they got different ideas about where the stem cells were. And we thought, well, maybe part of the problem is that everybody's looking at very small numbers of cells, and to really feel like you have the full picture, what you'd really like to be able to do is do high throughput confocal imaging of large segments of bone marrow and know where all the stem cells are. And so Malia Ajar and Malia Murphy figured out how to clear bones and bone marrow so that we could image the bone marrow within these cleared specimens. Here's an example of the data we get out. We have image processing tools that allow us to mask the cells we don't want to see so that we can see the cells we do want to see. And this allowed us to do three-dimensional digital reconstructions through the bone marrow. So I'm just going to play this movie just to illustrate our ability to fly through the bone marrow. Okay, so we can fly into the bone marrow. In what I'm going to show you is that the sinusoidal endothelial cells are green. The stem cells are green and white, and these leptin receptor positive niche cells are red. And so we can fly through the bone marrow and do close-ups on individual stem cells and image their interactions with the niche. Oh, sorry. I waited too long before I stopped it. So let me, this cell is likely to be a stem cell based on the markers that it expresses. The markers that we're using here are as good as the SLAM family markers or as any of the best markers in the world. And its position is typical for stem cells within the bone marrow. It's immediately adjacent to a sinusoidal blood vessel. And we've shown genetically, as I just showed you, that the key sources of factors for stem cell maintenance are these red leptin receptor positive cell and the endothelial cells. There are no stem cells that are not closely associated with blood vessels or with these leptin receptor positive cells. Now, it looks like these leptin receptor positives, the bone marrow is full of these leptin receptor positive cells. In fact, they're only 0.3% of the bone marrow. But as I'll show you in a minute, they have long processes that allow them to regulate the maintenance of many different cells within the bone marrow. So this is a close-up image. The original data is a much higher resolution than this, but my computer doesn't have enough computing power to run the original images. So more recently, Reto Fialko at UT Southwestern has developed a new way of doing lattice light sheet microscopy that allows us to image these bone marrow specimens at much, much higher resolution. Elise Jeffrey worked out a way to mark individual leptin receptor positive cells within the bone marrow. And so this is an image of a single leptin receptor positive cell. Here's the nucleus of the cell. Interacting, on average, they support the maintenance of 7 other hematopoietic stem cells and primitive hematopoietic progenitors. You can see what exquisite processes these cells have. So we're continuing to use a combination of genetic approaches and imaging approaches to image the interaction of these leptin receptor positive cells with the niche cells. So what I've told you so far then is that hematopoietic stem cells reside adjacent to sinusoidal blood vessels in the bone marrow, also in the spleen, another hematopoietic organ, and they derive their factors from these leptin receptor positive cells and from endothelial cells. So now that we've identified the location and the cellular composition of the niche, now we get to do the fun experiments to ask what else do these leptin receptor positive cells do. Bo Zhou came to my laboratory, one of the top postdocs along with Ray Yue, who's ever been in my lab. He's now got his own lab at the Chinese Academy of Sciences in Shanghai. And he showed that these leptin receptor positive cells are not just niche cells. They also include the skeletal stem cells that are the main source of new bone and fat cells that form in the bone marrow. These leptin receptor positive cells only represent 0.3% of cells within the bone marrow, but these are the source of the cells that give rise to the mesenchymal stem cells that can be cultured from the bone marrow. So what that means is that Bo could plate single leptin receptor positive cells from the bone marrow in culture, and those single cells could undergo multilineage differentiation to form bone and fat cells and chondrocytes. And that allowed us to both purify these cells and to fate map them in vivo for the first time. And Bo showed that these leptin receptor positive cells give rise to all of the adipocytes. Adipocytes accumulate throughout adulthood within the bone marrow. And they all come from these leptin receptor positive cells. And most of the new bone cells that form during adulthood also come from these leptin receptor positive cells. So you're constantly forming new bone cells that allow you to maintain skeletal bone mass as well as to repair fractures, for example. So what else do these leptin receptor positive cells do? Is there metabolic regulation in the niche? Are there undiscovered growth factors in the bone marrow? Could there be mechanical regulation? Now's our opportunity to really learn unexpected new things about the nature of the niche. So our first step in this regard came from Ray when he was in the lab. Ray did RNA-seq analysis in the leptin receptor positive cells to look and see whether they expressed any transcripts that would be predicted to encode secreted proteins whose function had never been studied in vivo. And he discovered a new bone-forming growth factor that's made by these leptin receptor positive cells. In fact, it's made by bone lineage cells and by a subset of the leptin receptor positive cells. And he showed, he called it osteolectin. Osteolectin acts on the leptin receptor positive cells to promote their differentiation into osteoblasts. Bo Shen subsequently came to the lab. Bo's still a postdoc in my lab, but he's got offers from universities in the United States and China to start his lab, and he'll do so in the fall. So he showed that the receptor for osteolectin is alpha-11 beta-1 integrin, and that when osteolectin binds to integrin alpha-11, it induces Wnt signaling in the leptin receptor positive cells, and that promotes bone formation. And if you delete either osteolectin or alpha-11, it leads to what looks like early onset osteoporosis, where you just can't maintain bone mass. So the story that I want to tell you now, the starting point for this story was asking whether or not osteolectin would be helpful as a marker to better understand the heterogeneity among leptin receptor positive cells within the bone marrow. So Bo knocked tomato into the osteolectin locus as a reporter to show us which cells were making osteolectin within the bone marrow. And he found that it was only 0.025% of bone marrow cells that were osteolectin positive. And those cells were all a subset of the leptin receptor positive cells. So not all the leptin receptor positive cells made osteolectin, only about 15% of the cells. And he cut sections through the bone marrow to figure out which leptin receptor positive cells were expressing it. So this is all bone marrow. This is the surface of the bone where the osteoblasts also make osteolectin. But within the bone marrow, he found that all of the osteolectin positive cells were clustered around arterioles within the bone marrow. And these other, so you can see it better here, these are the arterioles. And in a young mouse, they're all surrounded by these osteolectin positive, leptin receptor positive cells, where the sinusoidal blood vessels don't have any osteolectin positive cells around them. This was exciting to us because we knew from the papers that I already told you about that the job of these leptin receptor positive cells around the sinusoids was to make factors for stem cell maintenance. Ley had recognized that there were leptin receptor positive cells around the arterioles, but we had no idea what they did because we couldn't find any stem cells around the arterioles. So Bo used osteolectin as a marker to purify the periarteriolar cells and the perisinusoidal leptin receptor positive cells. And he did gene expression profiling, and he found that the periarteriolar leptin receptor positive cells had a gene expression profile that suggested they were poised to undergo osteogenesis, whereas the perisinusoidal cells had a gene expression profile that suggested they were poised to undergo adipogenesis. So that was consistent with data from 4 other labs that just in the last year or two published gene expression profile data that independently confirmed that these leptin receptor positive cells were the main source of factors for the maintenance of hematopoietic stem cells. And they also saw this functional difference between the leptin receptor positive cells, but nobody had a validated marker to functionally study these periarteriolar cells until Bo confirmed the osteolectin could distinguish these populations. So he knocked Cre-ER into the osteolectin locus, and that allows us to recombine conditional reporters. And that allowed us to fate map these cells. And so if we treat mice with tamoxifen to activate Cre-ER, it's only a subset of the leptin receptor positive cells that activate the reporter, and these cells are not very long lived. They disappear within 2 months. These cells disappear. So these cells are not stem cells. It turns out these cells are short-lived, rapidly dividing, restricted osteogenic progenitors. And they proliferate around the arterioles within the bone marrow. And then when they leave that microenvironment, the label traces into osteoblasts that are present at the surface of the bone. So this is a major population of new bone cells that helps to maintain the adult skeleton. Both the periarteriolar cells and the perisinusoidal cells in green express similar levels of stem cell factor, suggesting that the periarteriolar cells were also capable of creating a niche within the bone marrow. And so Bo used his Cre-ER allele to conditionally delete stem cell factor only from these rare osteolectin positive cells around the arterioles. And when he did that, there was no effect on stem cells and no effect on most other restricted progenitors within the bone marrow. But he saw a profound depletion of early lymphoid progenitors within the bone marrow. That suggested that there was a periarteriolar niche for the maintenance of lymphoid progenitors. So these are cells that make the B cells and T cells that are so important for immune function. So we imaged those early lymphoid progenitors, and consistent with what I just showed you, we found a subset of them, not all of them, but there's about 1/3 of them that are clustered around the arterioles within the bone marrow. And so we wondered what the physiological importance of this population is. And when we delete stem cell factor from these osteolectin positive cells, we see a depletion of early lymphoid progenitors, but there are so many homeostatic mechanisms in the hematopoietic system that even when you delete a progenitor population, there's ways of promoting the proliferation of other cells to maintain normal blood cell counts. And so the blood cell counts were normal in these mice, but when we stress the system by infecting the mice with a bacterial pathogen called Listeria, Listeria requires T and B cell function to clear. And so when you infect normal mice with Listeria, you see an expansion in the numbers of B cells and T cells in the spleen, shown in the blue bars. But if you inactivate this periarteriolar niche by deleting stem cell factor, now the mice can no longer expand the numbers of B cells and T cells. And as a consequence, they don't have the same capacity to clear the Listeria. This is a log scale. So there's almost 10 times as many bacteria in the spleens of the mice where we had inactivated this periarteriolar niche in the bone marrow. And the net result is that we could give the mice a dose of bacteria that only killed half of the wild type mice, but it would kill 90% of the mice in which we inactivated the periarteriolar niche. The other thing that Bo recognized was that these osteolectin positive cells became depleted as the mice got older. Now, most things in the bone marrow, including hematopoietic stem cells, don't get depleted during age. But it had been recognized for many years that these lymphoid progenitors did get depleted with age. You can see that these arterioles, in a young adult mouse, the arterioles look like this. They're all surrounded by these osteolectin positive cells. But in an old mouse, now you start seeing naked arterioles with no osteolectin positive cells around them. And you also see a depletion of lymphoid progenitors around these arterioles. So that suggested to Bo that maybe the reason why these lymphoid progenitors was depleted during aging is because their niche got depleted as well. And so he had what I think was a truly brilliant idea. He recognized that these osteolectin positive cells were bone forming cells, and that when mice or humans do load bearing exercise, they increase their production of bone cells. He decided to test whether he could rescue this decline of osteolectin positive cells in old mice by putting running wheels into the cage with the mice. So mice love to run, and if you just put a running wheel in their cage, they run all night long. And as expected, that caused the bones to get thinner. It increased the frequency of these osteolectin positive cells in the bone marrow. It didn't affect the frequency of stem cells or most other hematopoietic progenitors, but it rescued the decline in lymphoid progenitors during aging. So this suggested that this niche expanded in response to mechanical resistance. And this was really interesting because the way we understand mechanical resistance to increase bone cell production, the idea is that the mechanical resistance acts on the bone to increase the proliferation of osteoblasts associated with the bone. And this was raising the possibility that, in fact, those mechanical forces were being transmitted into the bone marrow and acting on these bone-forming progenitors inside the bone marrow. So Bo then tested whether mechanical resistance was necessary for the maintenance of this niche by tethering the tails of mice to pulleys on the lid of the cage so the hind limbs would be unloaded. So the mice could still walk around the cage on their forelimbs, and when they did that, nothing changed in their forelimbs, but their hind limbs, bones got thinner, the frequency of the osteolectin positive cells were depleted, there was no effect on stem cells or most other hematopoietic progenitors, but he saw a depletion of lymphoid progenitors. So this said, in fact, that mechanical stimulation is necessary for the maintenance of this periarteriolar niche. And in fact, these mice, if we inactivate this periarteriolar niche, they just don't have the same capacity to fight off infections anymore. So what's the mechanism by which these osteolectin positive cells were able to sense the mechanical stimulation? Well, Bo looked at their gene expression profiles and recognized that they expressed this mechanosensitive receptor called piezo-1 that was discovered by Ardem Patapoutian's laboratory. We purified these leptin receptor positive cells from the bone marrow by flow cytometry and did single patch clamp recordings to look at the channel activity, and we could see piezo-1. Piezo-1 is an ion channel that when the membrane of a cell gets stretched, the channel opens and it allows a current of cations to enter the cell. And so we could see functional piezo on the surface of these leptin receptor positive cells, and we could see it opening in response to mechanical stimulation. And if we just knocked out piezo-1 from these osteolectin positive cells, that was sufficient to cause the bones to get thinner, to deplete these osteolectin positive cells, and to deplete these lymphoid progenitors. And so these cells have to be able to sense the mechanical stimulation in their environment. And so what we think is happening is that, for example, if you go running, these arterioles go through the surface of the bone into the bone marrow. And when you go running, that movement-induced mechanical stimulation puts mechanical forces on the arterioles that causes them to stretch, and that transmits those mechanical forces deep into the bone marrow. And Mother Nature clustered these osteolectin positive cells around the arterioles and gave them piezo-1 to be able to sense those stretching forces. And those mechanical forces cause these osteolectin positive cells to proliferate, to form new bone cells that make bones stronger, but to also expand the niche for these lymphoid progenitors. And so we think that that this is part of the reason why exercise not only makes your bones stronger, but also improves immune function. So I'm just going to show you a handful of final slides to tell you one other short unpublished story. Joe, when he was in the laboratory, discovered that adipocytes that form after you ablate the bone marrow by either radiation or chemotherapy, the bone marrow fills up with adipocytes. And Bo discovered that that's an adaptive mechanism, that adipocytes are actually the emergency response system in hematopoiesis, and they secrete factors that help promote the regeneration of the bone marrow. And he showed that they make stem cell factor, but we had the sense that they probably do a lot of additional things. And so we've been studying what else do adipocytes do, and we realized that adipocytes make another growth factor called adiponectin. And adiponectin is known to be an anti-inflammatory growth factor. It's made by leptin receptor positive cells and adipocytes in the bone marrow. Now, other people have studied adiponectin in the hematopoietic system, and they showed that adiponectin does have effects on stem cells, but different studies observed different effects. Most studies found that it promoted the proliferation of the hematopoietic stem cells and promoted hematopoietic regeneration, but in other contexts, it looked like it was impairing the proliferation of restricted progenitors. So it was a little bit hard to tell from this literature exactly what adiponectin was doing. Is it positively regulating or negatively regulating stem cell function? What exactly does it do? So adiponectin receptors are broadly expressed by hematopoietic cells. And they're a better way of studying adiponectin function because the receptors actually can signal even when adiponectin is not bound. And adiponectin binding increases the signaling. And in other contexts, people have found that you get a stronger phenotype from studying the receptors than studying adiponectin itself. And so we knocked out the receptors in the hematopoietic system and found that when we did that, the hematopoietic stem cells, which are normally very quiescent, started dividing. And when hematopoietic stem cells divide, they mobilize from the bone marrow to the spleen to create what's called extramedullary hematopoiesis, where you start producing blood cells, not just in the bone marrow, but also in the spleen. This is something that happens when the when the hematopoietic system gets activated. So we wondered, in many cases, things that activate hematopoietic stem cells also exhaust them and lead to their depletion. And we saw the same thing with these adiponectin receptor deficient hematopoietic stem cells. So an assay that we do a lot is transplanting hematopoietic cells competitively into irradiated mice, and then looking at their capacity to produce blood cells over time. And in this case, we mixed normal wild type cells with the adiponectin receptor deficient cells, transplanted them into irradiated mice, and then we have separate markers in each cell type to monitor their contribution of blood cells over time. And the adiponectin receptor deficient cells had less function than the wild type cells. And if we serially transplanted these cells, the adiponectin receptor deficient cells became exhausted. And so that made clear that adiponectin receptor is one of the things that helps to maintain hematopoietic stem cells in the adult bone marrow. And it helps to maintain them by allowing them to stay quiescent and to not be activated by stimuli. So we wanted to know more about how this worked. And so we did the experiment a different way, where in this case, we mixed the cells together and we transplanted into the irradiated mice. And after we did the transplant, then we deleted the adiponectin receptor. And now when we did the experiment that way, there was no longer any effect of the adiponectin receptor. Now it looks puzzling at first, but to make a long story short, what I'm going to show you is that adiponectin receptors are not acting within the stem cells themselves. They're acting in other immune system cells, T cells and myeloid cells, to suppress inflammatory signals from those cells. And so if the stem cells come out of a mouse where adiponectin receptor was already deleted in the hematopoietic system, the hematopoietic system is very inflammatory. The stem cells get damaged and they get depleted. Whereas if you do the transplant first and then allow the system to get inflamed, the inflammatory factors are acting on both the adiponectin receptor deficient cells as well as the wild type cells. And so the adiponectin receptor deficient cells are not at a competitive disadvantage now in this setting. So this is what told us that the receptors were not acting in the stem cells themselves. So when we tried to understand more about this inflammatory environment, we could see that the adiponectin receptor deficient immune cells were making high levels of lots of different inflammatory growth factors, creating a very inflammatory environment that activates and depletes the stem cells. And so we could protect the stem cells from this environment by deleting the receptor for interferon, one of these inflammatory factors. So normally, if you delete the adiponectin receptors from hematopoietic cells, the stem cells go into cycle. But if we delete interferon receptor from, we partially rescue them going into cycle, and we partially rescue the depletion of the stem cells as well. So it's this inflammatory environment that's promoting the depletion of the stem cells. And it's not just interferons, it's also another inflammatory factor called TNF-alpha that's contributing to this effect. And so if we looked in the context of aging, in the absence of these adiponectin receptors, the stem cells don't persist into aging. You have to maintain hematopoietic stem cells in your bone marrow for your entire life because you have to be able to produce new blood cells throughout your entire life. And you depend on adiponectin receptors in the hematopoietic system in order to do that. Because otherwise, every time you get an infection, the system overreacts, becomes highly inflammatory, causes the stem cells to divide inappropriately, and that ultimately leads to the depletion of the cells. And if we transplant these cells from old mice, the stem cells become depleted where the wild type normal cells still have the capacity to make blood cells. So just to sum up this final piece, leptin receptor positive cells and adipocytes make adiponectin. That adiponectin is acting on T cells and myeloid cells within the bone marrow to suppress their capacity to make pro-inflammatory factors. And that helps to protect the stem cells. But if we delete adiponectin or adiponectin receptors, we increase the production of pro-inflammatory factors. We cause these stem cells to start dividing, and that leads to extramedullary hematopoiesis and the depletion of stem cells with age. And so the conceptually important point here is that the niche is not just making hematopoietic growth factors, and it's not just regulating mechanical stimulation within the bone marrow. It's also protecting the stem cells from the immune system, which otherwise creates an over-inflamed environment that prevents the stem cells from persisting throughout adult life. And so the point is that this is an interesting frontier in stem cell biology where these microenvironments that maintain the cells that maintain our tissues are doing lots of different things to regulate tissue homeostasis. So I'll stop there, and I mentioned the people all the way through, but for the two last stories that I told you, the story from Bo Shen about the mechanical stimulation was just published a couple of months ago in Nature. The data that I showed you on adiponectin hasn't been published yet. It's from Corbin Meacham in the lab. Obviously, many people contributed to this work over the years in my lab, including Ray Yue, who's there at the meeting, and now Vice Dean, Department Chair at Tongji University, and Bo Zhou, whose work was really important for this story that I told you, who's at the Chinese Academy of Sciences in Shanghai. So I'll stop there. I'm not sure if we're taking questions, but I'd be happy to answer any if there's time.
Thank you, Sean, for your terrific talk. So if we have some questions, please raise your hand. This, Hi, Sean, can you hear the question?
I can.
Oh, very nice talk. And I have two questions about the OLN positive cells and common lymphoid progenitors part. First one is that whether extra administration of the SCF could rescue the common lymphoid progenitor defect in the aging mice or in the OLN CRE-ER SF flux mice.
It's a really good question. And we haven't done the experiment because it's hard to get enough stem cell factor to inject. And these effects, become apparent over long periods of time in the mice. So it's a hard experiment to do. The other thing to think about is that there are two forms of stem cell factor. There's a membrane bound form and there's a soluble form. And we know the membrane bound form is important. The soluble form may be important as well. And so it's actually one of the interesting questions now to understand the relative contribution of soluble stem cell factor versus membrane bound stem cell factor. And it's something that we and other people are working on.
Oh, thank you. And the second question is we know the running or the tail handing leads to the different central neural system, neural activity. So have you ever considered that whether this central nervous system neuron activity difference leads to the different behavior in the OLN cells, for they could transmit this activity through the neuron near the bone marrow and directly function in the OLN positive cells?
Yeah, it's a really great question. There are nerve fibers from the peripheral nervous system, so sensory nerve fibers and sympathetic nerve fibers that innervate the bone marrow. And they do regulate the regeneration of the bone marrow, as well as various aspects of stem cell function. Other laboratories have shown that those nerve fibers are important for regulating certain aspects of stem cell function. And we're now studying the interaction between the niche and those nerve fibers.
Any more questions from the audience? If no further questions, let's thank Sean for the great talk. So hope you have a good night.
Thank you for the opportunity, and congratulations again to all of the Ray Wu Prize winners. And congratulations on being able to have a big symposium like this. It's fun to give a talk to a live audience. I just wish I could be there in person with you.
Thank you. Bye.
Thank you.
Bye.